Okay, so this is general biology, Bio131, and we're going to start chapter 6. And chapter 6 is about the DNA molecule. And we are going to cover most of these subjects, not all of them, but most of them. We were going to talk about all aspects of DNA, including replication, including making RNA out of DNA, which is transcription, and we talked a little bit about that before, but we will have some details to talk about, including translation, gene expression, and mutation that can happen, and then cancer and genome mapping at the end. So if you start with all of these organisms, they do not have the same genetic code, but it's very similar, even though they are different organisms, different things, but a very good part of it is shared between humans and animals and plants and so on. So if we go to the molecule of DNA, DNA is a short of or abbreviation of deoxyribose nucleic acid. Deoxyribose nucleic acid. What does it mean? It's a nucleic acid that have a sugar called deoxyribose. How about RNA? It's a nucleic acid that have ribose. So what's the difference? Ribose and deoxyribose. Ribose is a sugar that have 5 carbons. Deoxyribose is the same one. You just remove oxygen. Is that okay? Deoxyribo nucleic acid, a nucleic acid that have deoxyribose, plus other things that we will discuss, but this is where the name came from. And as we should already know, the DNA is a double helix. It's 2 helix. If you look at this here is one, and here is the other one. So you have 2. It's a double helix, and each one strand, so it's 2 double helix or 2 strands, and each strand is a big chain, a big string of nucleotides. We will go to the details. So follow with me as we go. Do you remember in the very beginning when we talked about the chromosomes, right? Chromosomes, DNA, surrounded. You put the DNA and you make the chromosomes, chromatin chromosomes, and so on. So the chromosome is a double helix. So it's a double strand. Each strand is a string, a big chain of nucleotides, and we will discuss the details. But follow with me as we go. So I'm going to look at this long chain of nucleotide. Okay? So we call it polynucleotide. Polynucleotide. What's poly means?
More than one.
More than one, many. So polynucleotide, many nucleotides, a long chain of nucleotide. What is the nucleotide? We have a long chain of it. What is it? It is a base with sugar and phosphate. So you have nucleotide, another nucleotide, another nucleotide. You make a chain of nucleotide, and I call that polynucleotide. This is one strand. If you put the other strand, that will be DNA. And do you guys remember how the 2 strands stay together? How do they stay together? There is a hydrogen bond between them, and the bases fit together somehow that I'm going to discuss now. But generally speaking, here is the specific pairing, base pairing. So we have 4 bases, right? So again, the DNA is 2 strands. 2 strands of what? Polynucleotide. What's a polynucleotide? A big chain of nucleotide. What's a nucleotide? Base, sugar, and phosphate. I'll start with the base. The bases are 4 bases. A, T, C, G. At, C, G. A, T, C, G. A is for adenine, T is for thymine, G is for guanine, and C is for cytosine, or the other way around. ATCG. The two strands that we're talking about, we say it's a double strand. These two strands are called complementary strands. One strand and the other one will complement. So both together will be DNA. You need to notice something here, that the reason why we always say ATCG in that sequence, ATCG, at CG. Why? Because the A fits with the T only. So and the C fits with the G. So if I have A in this strand and T in this strand, they will fit together. C will fit with the G. Okay? And the simple reason why is, look at this, for example. If I have this strand right here, this is the A and this is the T. Okay? And let's say this is the C and this is the G. Like this. See, this will fit with this. This will fit with this only. But let's say this is a triangle out, and this will fit in this triangle. This is AT, CG. Do you think the C can fit with the T, for example? No, this is a square and this is a triangle. So this is how it fits, and this is how you should remember ATCG. A with the T, C with the G. So if we have a sequence here, and this is a double strand, both of them, and these are the two complementary strands. So if this one have ATCACGC, can you come up with the complementary strand if you didn't see this?
Yes.
What's here?
T.
And next?
A.
Next?
G. T. G. C. G.
Okay. So this is how you can make up the other strands. You can see a question like this. I can ask you, true or false, this fits with this. Or which of the following is a complementary strand of this? So you need to find out which is the right one. So if I give you half, you should be able to figure out the other half. If you know half, you know the other one. And replication. Replication is something that's completely different than transcription and translation. We have all these topics that we'll talk about. But replication means you make DNA out of DNA. Okay, which is needed obviously in the case of mitosis. Do you remember the mitosis? We talked about it last week. So if that's the case, you're duplicating the DNA. So I make DNA out of DNA. This is completely different than transcription when I make RNA from DNA, right? DNA from DNA. What do you call it? Replication. You're replicating. You're duplicating. Okay? So replication is the first thing. I have a double strand of DNA, and I will make a double strand of DNA that's exactly the same. You're duplicating the amount. Okay? So what you're going to do is, since the two strands are coming together, like it's zipping together, zip together, so, and of course A with the T, C with the G, and so on, right? So the two strands are together like this. And you need to unwind it first, untwist, and then separate. Okay, we call that unzipping. Okay, you separate them into two. Now I have, here is one, and here is one, separate. Okay, each one of those is going to make another one that is complementary to it. Okay, so let's say this is 1 and this is 2. 1 will make a complementary strand. Now it's a double strand. 2 will make a complementary strand. Let's say 3 and 4. 1 with 3, 2 with 4. So now I have two double strands. So now I doubled the amount of DNA that I have, right? Did you get this? This is replication. Replication. And we call the replication semi-conservative. Semi-conservative. Why? Because I have the first pair, we call that the original one, right?
Yeah.
This is the original one. When I unzip it and separate into two, this is half and this is half, right? So you're going to make a complementary to this half. So this looks like this half, and the other one looks like this half. So we call it semi-conservative. Why? Because it's half conservative. You did not make the new strands out of the blue. You didn't make it out of nothing. You make it out of half of the DNA that you have. Did you get the idea? You have 2 strands. I will use those as originals and make copies of those. So we call this process semi-conservative. It's half conservative because half of it is coming from the origin. We call the 2 strands that are newly formed, we call the first one that you make a copy of in order to make a complete DNA, we call that the parental strand. And the other half is complementary to it. So that's why we call the DNA semi-conservative, because you have 1 strand original, okay, and 1 strand new. So this is called semi-conservative or semi-conservative. Okay. To describe the details of the replication process, I have a short video here. You can, let's watch it together, and we'll understand the process.
DNA is a molecule made up of 2 strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The 2 strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand, and wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The 2 strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the 2 strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adding bases 1 by 1 in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
Okay, so did we understand this process? you guys, I know it's not in this slide, but you need to remember DNA polymerase. This is the enzyme that help you to make the replication, make DNA out of DNA. Helicase is the one that separate the helix from each other. It's the enzyme that separate the helix from each other. Okay. We understood the leading strand and the lagging strand. The leading strand is the one that moves straight continuously, and the lagging one that's pieces, and then you're going to put them together. And we understand that it's semi-conservative. These are the things, the details that you guys need to know. ATCG, these are the building blocks. And this is what's needed to make the DNA. If you think about these the way you put them together in different directions, ATCG, ACG, CTC, whatever the direction is, there are thousands of ways that you can put them together. And it's, if you unfold it and put it together, that's in a nucleus, it will make a extremely long strand. So this is how long it is. It's just all these repeats of 4 bases. Okay. So if I ask you what is the structure of the DNA, 2 strands of poly what? It's a 2 strands of what? Polynucleotide. Each strand consists of polynucleotide, which means several nucleotides. What's a nucleotide? Base, sugar, and phosphate. What kind of sugar? Deoxyribose or ribose? Deoxyribose. This is a type of sugar. And what kind of bases? 4 bases. What are the 4 bases? ATCG. Did you get this so far before moving to the RNA? Okay. Now the RNA. RNA is another nucleic acid. What do you mean by nucleic acid? It's an acid in the nucleus. But this one contains ribose instead of deoxyribose. So instead of DNA, it's RNA. Other than that, there are other differences between the DNA and RNA. So the first difference that we mentioned is that this one contains deoxyribose and this one contains ribose. Is that okay? This is for the sugar. How about the DNA being a double strand? RNA is not a double strand. RNA is a single strand. This is number 2. Number 3, what are the 4 bases in the DNA? A, T, C, G. And instead of that, you have AUG. The RNA does not have T. It have U instead. Did you get the differences between the DNA and RNA? Which one is single strand? RNA. And which one is double strand? DNA. Which one contain ribose? RNA. Which one contain deoxyribose? DNA. Which one is ATCG?
DNA.
And which one is AUCG? This is RNA. Okay? So what's a DNA in the first place? DNA is the original script that contains the basic information in order to make any type of protein. If you wanted to make a protein, you go to the DNA and tell the DNA, I want to make this type of protein. Give me the script. Give me a copy of that script so I can make it. So all the information that's needed to make a protein is on the DNA. But we talked about this before. Do you guys remember when we talked about the nucleus, that the DNA cannot leave?
Yeah.
Why cannot leave?
Too big.
It's double strand too big, right? RNA can leave the nucleus, right? Small enough to pass through the nuclear pores. You still remember this? So what's going to happen is, I want to make protein. Where are you going to make the protein? In the factory. What's a factory? Huh? Rough endoplasmic reticulum. Which part of rough endoplasmic reticulum? Ribosomes. And where is the ribosomes? Is it in the nucleus or in the cytoplasm? In the cytoplasm. So I want to make protein in the factories. And the factories are in the cytoplasm. But the information is locked inside the office of the manager. I need to make a copy, and this copy can exit the office of the manager, which is the nucleus, in order to go to the cytoplasm and go to the ribosomes, so you can make the protein there. And in order to do that, you need to make a copy of DNA that's called RNA. And what do we call this process of making RNA out of DNA? Transcription. When you transcribe something, means you make a copy of. Transcription. So we're going to make transcription, which is RNA from DNA, and then that transcript, remember that the transcript is not a copy of the whole DNA. It's a copy of part of the DNA that you need, that have the information for that specific protein. Did you get the idea? Are you going to make a copy of the whole thing? No. Because the DNA contains information about every single protein. Are you going to make every single protein every single day? Of course not. Today I need to do this one protein or two or something. So you're going to take a copy as much as you need. Did you get the idea? You go to the DNA. How many transcript do you have? I have like 30, 24. I need 2 of those only. I want to make 2 proteins only. So I'm going to make a copy of this part only that have those 2. Did you get the idea? This is how it goes. So the RNA is a copy of DNA that have the information for what you need to do only. Okay? And then the messenger RNA is going to go to the ribosomes to make proteins. Why do you make proteins? For a lot of things that I talked about that before, if you still remember. It may make part of the membrane, it make part of different structures, it can be secretion. Do we still remember these? Hormones, whatever. A lot of uses for proteins. Okay, so this is how it goes. It's in a nucleus. Chromosomes are in the nucleus, which is DNA. You're going to make RNA, and RNA go to the cytoplasm, where it finds the ribosomes. All ribosomes are located in the endoplasmic reticulum.
No, there's some scattered in the
Some scattered. What do you call it?
free
ribosomes. So there are fixed ribosomes and free ribosomes. Fixed ribosomes, fixed on what?
on the rough endoplasmic.
On the rough endoplasmic reticulum. And that's why it's rough. So if you think about it, from the beginning to the end, I have information stored for making everything. Okay? Every single protein that we can make in the body, the instructions are located inside the DNA. So if you think about the color of your eye, the color of your eyes is a pigment, isn't it? The iris, do you know the iris? The colored part of your eye is called the iris. This iris can be of different colors. And these colors are because of pigments, which are initiated by a protein. So protein is the one that control the color. So if you think about it, from the DNA until you get to the color of the eye, which is the physical pigment, think about it. It will go DNA in the nucleus. You're going to unfold it and make a small copy, which is RNA, that's leaving the nucleus through the nuclear pores and go to the ribosomes. And the ribosomes, you're making that protein, and that protein is going to the eye, if that's the case, to the iris, I mean, and it's going to direct the creation of that pigment. So here it is again, DNA to RNA. What do you call this? Transcription. And RNA, it can leave, and then it will go to the ribosomes from which you're going to make the protein. So transcription is you're going to make RNA from DNA, and the base pairing in this case is you're going to use U. We discussed the differences between RNA and DNA, right? So if the DNA have an A, what's a complementary strand in the RNA? Huh? U. Isn't supposed to be AT, CG? AU in RNA. So when the RNA see A, it's going to make U, not T. Okay? And this will make transcription is when we make messenger RNA out of DNA. Okay? Is that okay for that transcription? You're making messenger RNA out of DNA, and the RNA is complementary to the DNA. Understand this. You're making a copy, but not the exact copy. It's a complementary copy. Do you remember the old-fashioned cameras when you have this negative, right? Like your hair is black, you see it in the negative white, right? If your color is dark, you see it white. If it is light, you see it dark, right? It's the negative. We call it the negative. Did you see that? The old-fashioned ones? This is exactly the RNA. So it doesn't look exactly like the DNA. It's complementary to the DNA. So if this is a DNA ATCG, the RNA is not going to be ATCG. Okay? It's going to be AGC. Did you get the idea? It's not a copy, it's a complementary copy, like a negative of it. And you're doing that because when you read it, you get the original again, just like the DNA. Like when you take that negative and print it, does the picture looks like you again? It does, right? But you're converting the actual shape, your face, into a negative, or the picture into a negative, and this negative, when you print it, it will look like the original, right? This is exactly what's happening. RNA is a complementary of DNA, and then when you make protein, the protein will look exactly like the original copy that was in the DNA. Okay? So this will bring us to the next part, which is called the translation. And the translation is ribosomes are translating the message on the messenger RNA. The ribosome is two subunits, like this is a ribosome, small unit and big unit. Here's what's happening. This is ribosome. That's not doing anything. Then the messenger RNA came. When it comes, they are going to separate from each other. And you put the messenger RNA in between, and you do like this. Okay, you put it here, and you put the two units again with the messenger RNA in between. So we're doing that in order to translate. You read and translate. You read it as triplets, which is called the codon. Are we following? Each three bases are called codon. So ACU, this is a codon. UCC, another codon. GCU, this is another codon. Okay? So in the messenger RNA, what do you call it? Each triplet, codon. Is that okay so far? So our ribosomes are going to read, but when it starts to translate, it will translate it into the original ones. Okay? And you're going to end up making amino acids for each. So what's a codon for? What does it represent? 3 pairs that tells you which amino acid to make. So the codon represent what? Amino acids. Did you understand this? Each triplet is called a codon, and each one codon represent or help you to make one amino acid. Is that okay? So if I have let's say 30 codons, how many amino acids are you making out of those? 10 amino acids. Okay? So you read the first 3 amino acids, go to the next amino acid, third, fourth, and every 3 will make you amino acid. Out of the 30, I'll get 10 amino acids, and then I will link them together. These amino acids, I will link them together, and we call that the link in between each 2 amino acid is called a peptide bond. So you end up making a polypeptide. Did you understand? You're reading, each 3 is a codon, and each codon will be translated into amino acid, and that's why we call this process translation. You're translating the codon into amino acid. Okay, so you're reading 3 codon, amino acid number 1. Next 3 amino acid number 2. Now, are you going to have just amino acids separate? No. When you make amino acid number 1, when you make number 2, you link them together. What's the link? A peptide bond. A peptide bond. Okay? Then you make number 2, and you go to the third one. Show me the codon. Translate into amino acids. And then you link it to number 2. And then you make 4, link it to 3. Until you have a long chain, and we call that polypeptide. Is that okay so far? Did you get this starting from a transcription until you make a polypeptide? Okay, polypeptide. I didn't say protein. I said polypeptide for a reason. You don't call it protein yet. Are we still following? You call it a polypeptide, not a protein yet. When you have just a chain of amino acids, 1, 2, 3, 4, 5, 10, whatever it is, you just call it polypeptide. It's not a protein yet. If you want to make it a protein, you need to do other things. Okay? You're going to fold it around, or you twist it around, and then you're going to fold it, and then you make different parts, and you put all of that together to make a protein. So polypeptide, you make protein out of polypeptide. Okay? What is the gene? The gene is a code, a part of the chromosome that help you to inherit something. So this gene is for the color of the eye, this gene is for the color of the hair, and so on. So it's part of the chromosome that help you to inherit something. Okay? This is the transcription again. You're making RNA out of DNA. And by the way, the first part is the same. Do you remember when I showed you that video? What do you do the first thing? Unzip it, right? You unzip it first. You're doing the same thing here. And then you're making a copy of DNA. But this time it's an RNA. The copy is RNA, right? So what was the enzyme that was helping you to make DNA out of DNA and replication? What is the name of the enzyme that help you for replication, to make DNA out of DNA? DNA polymerase. DNA polymerase. Okay? DNA polymerase. Polymerise. Polymerase. Okay? Polymerase. How about the enzyme that help you to make RNA out of DNA? RNA polymerase. Is that okay? What do you call the enzyme of replication?
Polymerase.
Which polymerase? Both of them are called polymerase. DNA polymerase. And what do you call the enzyme that help you with the transcription? RNA polymerase. Okay? Here is another video that's describing this process of transcription. It's a short video. Let's just see it. Let's watch this. So it sticks more and should be able to remember.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called a nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
Okay, did we imagine the whole process? Starting with the DNA until you end up having a protein. And I mentioned that polypeptide is not a protein. Did you see it? You have to fold it and make a 3D structure, and now you call it a DNA. And I didn't talk about the transfer RNA, and this is the third type of RNA. And transfer RNA, from the name, is transferring the amino acids. This is the one that fits into the messenger RNA. And what did we call it? Each 3 bases in the messenger RNA? Each 3, what do you call it? Like ACG, huh?
Codon.
We call it codon, right? You call it codon. Now the question is, how the transfer RNA know that this is the right amino acid? Like, if it fits in. Okay, you have AUG. The transfer RNA will come. Okay, let me fit in with you. Okay, so let's say it's ACG, it will be UGC. Yes, we fit, so this is the right amino acid. I will leave it and go. And then you read the next 3, which is a codon. The transfer RNA have an anticodon. Codon and anticodon, they fit together. If it fits, that's okay. The transfer RNA is going to leave the amino acid and you link it together, the 2 amino acids. What do you call that link? Peptide bond. Peptide bond, right? Third one, you read the triplets, which is a codon. And what is the correspondent to the triplet in the transfer RNA? Codon is on the messenger RNA. How about that on a transfer RNA that fits with it? Anticodon. Anticodon. The triplet, the 3 on the messenger RNA, codon. The 3 on the transfer RNA that fits with the codon, anticodon. Is that clear?
Yes.
Okay. So anticodon, another amino acid, link the amino acids together to make a polypeptide bond. Is that a protein yet? No, it's just a chain it's a polypeptide, a chain of amino acid. Then you're going to twist, fold, you make a 3D structure, and you call it a protein. So what's the gene again? It's a stretch of DNA that produce one protein. So what's the difference between gene and codon? The gene have instructions for one protein. The codon have instruction for one amino acid. So obviously the gene have a lot of codons. Did we understand?
Mm-hmm.
Did you get the idea? So here is the DNA. Let's say this is the DNA. Protein number 1, protein number 2, protein number 3. I'm going to make protein number 1. The whole message, the whole thing. This is called the gene. Gene for protein. If you look inside the genes, you see codon, 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 codon. Which one will correspond to? Amino acid. Did you get the idea? The gene represents one protein. The codon presents one amino acid. Okay? Now if you wanted to study the DNA, like some people do personal DNA test, do you want to do it or not? Do you think we should do it or not? If this is a lady or parents that are having a baby, should you do it to the baby or not? If she's pregnant, should you do it during pregnancy or not? What do you think? Would you do a DNA testing or not? Personal DNA testing. Everybody would be different, but what are the benefits? Why would you do it? Like for yourself, if you make DNA for yourself, DNA test, what can you use it for? Why would you do it? Huh?
Possible sequences.
Yes. If you do a testing for the DNA, you might at least expect what's going to happen later on. Okay, they tell you that this is happening here, and you might end up having this issue. This is one. If this is a pregnant lady, that the fetus is having an issue, they do that. This is like most of the pregnant people that are older, like older than 35 or 40, they usually do it. They get a sample from the amniotic fluid, which is the fluid surrounding the fetus. They take a sample of that by a needle, and they take some of the cells and study the chromosomes and the DNA, and they can actually tell. It can diagnose a lot of things. This baby is going to have a spina bifida. This baby is going to have, when he is born, he will have cystic fibrosis, for example. They can tell.
You mean like mutations?
Mutations, yes. You can find the mutations in the amniotic fluid. And even if after birth, you can take some cells. They usually take the cells from the mouth. Just a swab like this from the mouth, and you will get a couple of cells, and they study that, and they can tell them what's going to happen, or what's most likely going to happen. Okay, so this is a translation again from DNA to messenger RNA to transfer RNA. How many RNA do we have? 2 or 3? There are 3. What's the ones that you have? Tell me what do you know about RNA? How many types? Or which types do you know?
Ribosomal.
Ribosomal is one.
Messenger.
Messenger. Transfer. So there is a ribosomal DNA. RNA, I mean. Yes. The messenger is the one that we mentioned, transcription, you make a messenger. Is that okay? Mm-hmm. The transfer is the one that help you to transfer the amino acids. How about the third one? The third one is ribosomal RNA plus proteins. We call that ribosomes. So the third one is called ribosomal RNA. What's the difference between ribosomal RNA and ribosome? The ribosome is ribosomal RNA plus some proteins. Did we understand this? Mm-hmm. So what are the 3 types? Messenger RNA, transfer RNA, and what's the third one? Ribosomal RNA. Where is the ribosomal RNA? In the ribosomes, right? It's in the ribosomes. So what's a ribosome? It's a ribosomal RNA plus proteins. Is that clear? So this is a little bit of details of what's happening there. Here is what I just mentioned. The tRNA, the triplet is called, what do you call the triplet on the transfer? Anticodon. Anticodon. What did you call the triplet in the messenger RNA? Codon. So the anticodon of the tRNA is going to fit in with the codon of the messenger RNA. And where is that happening? This matching happening? It's happening in the ribosomes. Okay? If they fit, Here is the triplets. Look at this. 1, 2, 3. So let's say ACG. And this triplet, which is the anticodon, the green is the anticodon, the red is the codon, okay? If the green fits in with this, like UGC, if it fits in, yes, this is correct, and I'll let this amino acid go. Okay? This like something like a map that help you to know what the code means. Of course, you don't need to memorize this. This is too much. Okay? But just look at this. Understand how to read it. Are we following? If you see something like this, at least you should be able to read it. So let's start from here. A. Okay? I'll start from A. A, it can be followed by G or A or C or U. Let's take it one by one. AG. AGG. This is alanine. Arginine, I mean. Arginine. AGA. Also arginine. So do you mean that I can have 2 codes for the same amino acid? You can have even more than 2. You can have even 3. Okay? So let's go to the next one. AGA. Same thing, right? How about this? AGC. Serine. How about AGU? Also serine. Okay? So you can have more than several codes, and all of them will mean the same thing. Look at this one here. UCU, serine. UCC, serine again. UCA, serine. UCG, serine. How many codes? 4 codes. 4 codes. That's okay. If you see any one of those, it's the same thing to me. I will give you the same amino acid anyway. But did you get the idea of how to read this? You start from the center. Yes. What is it? Start sign?
Stop sign.
Stop sign, yeah. This is a good question. When you're reading the messenger RNA, this is a messenger RNA, right? It contains triplets. Triplet, 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 triplet. Each one of those triplets is called a codon, right?
Mm-hmm.
This big strand of messenger RNA, it will tell you exactly when to start and when to stop. You're not going to go forever, okay? So there is one code or more than one code that translated into start. A codon that means start. Yes, not all codons are translated into amino acids. There are a couple of codons that means start and a couple of codons that means stop. When you see this code, this is where you start. When you see that code, you're done. Stop. Okay? So look at this here. UGA or UGG. UGA. It means stop. Once you see it, you're done. Let's say you got into 55 amino acids. Should I continue going? When you see this sign, it will tell you you're done. That's it. Right? Like if you have instructions for something, and at the end it will say the end. You're done. You're done. Okay? So this is a start sign, and there is also the stop sign. So these stop signs are UGA and UGG, and here's UAG or A. Okay? When you see any one of those signals, it means stop. You're done. There is other signals that means start, like AUG, for example. It's not showing in this, but this is what it means. AUG means start. So if you look at this, can you tell me what are these amino acids?
A little bit.
Just look one by one. Did you get the first one? Read the first one. Okay. What does it mean? Methionine. Methionine. Okay. Next. ACU. Can you read it? ACU. Threonine. Right? AAA. What does it mean? AAA. Lysine. Yes. Okay. So did you get how to read it at least?
Yes.
I'm not going to ask you to remember or memorize the codes, but you have to know how to read it. Okay? So when it comes to the translation, when you translate, you have initiation, elongation, termination. Okay? This was in the video. I showed you that video for translation. Initiation, this is when you start. Do you remember this? When you initiate and you start making the amino acids, and then you add one after one after one after one, and when you see the stop sign, this is where you start the termination. So when are you going to initiate? When I see start sign. The start sign is AUG. Okay, and then elongate. I will continue. Elongate more until I see the stop sign, which is WAA, WAG, WGA. Any one of those, it means stop, and this is where you terminate. And when you terminate, the two units are going to separate. Messenger RNA leaves. You go back to when you started. Okay? At the end, you end up having a polypeptide. When you see the stop codon, you're done. You didn't make a protein. You make your polypeptide. You're done. This is the last amino acid that you need to make, and you're done. So when you see it, the ribosomes are going to split into the two parts again, releasing the messenger RNA and releasing the polypeptide. The polypeptide need to be modified and changing the shape, twisting it, okay, folding it until you get the 3D shape of protein. So the protein is a 3-dimensional shape, okay, of amino acids. And where do you think it will go after that? You can use it as it is, but you can also modify and export it out. Who is doing that for you?
Golgi apparatus.
Golgi apparatus. You can send it to the Golgi apparatus, and the Golgi apparatus will take it from there if you want it to export, right? Send it to the Golgi apparatus. You can't use it as it is. You need to send it there, modify, package it, make it nice and ready to export, and then kick it out, which is called exocytosis. But it has to be ready for export. Okay? Okay, so this is good enough for this part, and we'll do the next part next time.